In this lecture, we'll go over the first part of the chapter on cell division. We won't go over everything in this outline. We will focus on the first few topics. We'll talk about why do cells divide, then we'll talk about the process of cell division in prokaryotes or bacteria. And this is fairly simple because the chromosomes in bacteria are pretty simple in their organization. We'll contrast the organization of the DNA in prokaryotes with that of eukaryotes. Eukaryotes have much more complexity in the DNA, both in the amount of material, but also in having many linear chromosomes instead of one circular chromosome. And so this will make the process of cell division more challenging. And then we'll talk about an overview of the cell cycle, especially as it applies to eukaryotes. In subsequent lectures, we can talk about the remainder of this chapter. So one important idea in cell division is that every time cells divide, there must also be a DNA replication or duplication event. Because each of those daughter cells will need its own copy of the DNA, then sometime before the division, that DNA must have been copied. Then, during the process of cell division, there has to be some process that makes sure one copy of the DNA ends up with one daughter cell, while the other copy of the DNA ends up with the other daughter cell. And cells need to divide not only the DNA. The DNA is usually what we focus on, but the cytoplasm has to divide as well. There are many organelles that will need to end up with one copy in one daughter cell and the other copy in the other. And so we'll talk about that process, which is cytokinesis. We will go over to start with prokaryotic cell division because it is pretty straightforward. Here's a table that you should be able to fill in by the end of this chapter, and this will be one way of knowing whether you understand everything that's happening in this chapter. So we can divide two processes. One is binary fission, and the other is mitosis. You should know which kind of organism does both processes. So binary fission is prokaryotes, and mitosis happens in eukaryotes. You should know the shape of the chromosomes in both, with circular shapes in prokaryotes and linear chromosomes in eukaryotes. You need to know about the timing of DNA replication compared to DNA division. We'll see that in prokaryotes, these events happen simultaneously. In eukaryotes, we'll see that DNA replication happens prior to DNA division. It replicates first, and then later it gets divided. We'll see that there are a few different kinds of methods by which the cytoplasm divides. And we'll see a process called septation in bacteria. We'll see a process either called a cleavage furrow in some kinds of eukaryotes, such as animals. And we'll see formation of a cell plate in other kinds of eukaryotes, such as plants. Because there's different ways of dividing the cytoplasm, there's also different names for the walls that are formed. We'll see that a septum is formed in bacteria. We'll see that a cell plate is formed in plants, and this doesn't really apply, as we'll see when animals divide. There is a cleavage furrow that ends up causing two different cells, but the cleavage furrow isn't really something that remains behind afterwards. I think that will be more clear when we get there. So going over prokaryotic cell division, remember that the process is binary fission. And remember that binary fission is not the same as mitosis, different processes. This is just a picture of some bacteria to remind you that's what we're talking about. And in prokaryotic cell division, we'll see that the one chromosome gets copied and separated into the two daughter cells, and then a wall called a septum forms. The DNA in bacteria is one circular chromosome. That's a little bit of a lie. There are bacteria that have multiple, but we'll stick with the most common situation, which is what your book focuses on. and that is having one circular chromosome. And what we are going to see is that division of the chromosome also results in separation of the two chromosomes. And I think we can see that more clearly on an upcoming slide. No, it's right here is the best place to look at it. And so we see two areas here. This area in green is the origin of replication. So we now have the origin of replication labeled here. We will see that in bacterial DNA replication, it is going to happen in both directions simultaneously. So it does not just happen going clockwise or just happen going counterclockwise. It's happening both ways. The result of this is that we get a second chromosome forming. In this picture, it's happening on top of the first one. I will see here if I can switch over to a GIF that shows this more clearly. And here you can see a GIF that is showing this process happen, where we have an original chromosome that is half red and half blue, and it copies itself. As it copies itself, the blue copy separates off to the right-hand side until eventually the two copies are completely separate. So this is what we mean when we say that in prokaryotes, the process of DNA replication and the process of DNA division are simultaneous. It's being replicated, and then as it's replicated, it is also dividing into a separate chromosome. Because it is proceeding in both directions simultaneously, eventually in both directions, the process will end at this termination site or terminus at the opposite end of the chromosome. At this point, the two chromosomes can entirely separate. And we can see that here, once they are separate structures, they can move to two separate sides of the cell. Now notice that the DNA is still double-stranded. That is true of DNA in both prokaryotes and eukaryotes. Now we need the formation of a cell wall between the two daughter cells. This process is going to happen by the process of septation in prokaryotes, so the formation of a wall. The septum is also the term for the divider between your two nostrils. It simply means a divider. So the septum forms, septation is the process that forms it. That sums up what we want to know about prokaryotic cell division. Things are much more complicated in eukaryotes. Why is that? Well, eukaryotes have multiple chromosomes, so they need to worry not just about replicating and dividing the one, but they have to make sure that all of the others have one copy that goes to one daughter cell and one copy to the other daughter cell. Additionally, eukaryotes have internal membranes. So the nucleus is enclosed by a membrane. And then there's also the rough and smooth ER and the Golgi apparatus, as well as potentially things like vacuoles. These all have to get divided and have one organelle end up in each of the daughter cells. So in a eukaryotic cell, for every chromosome that is in the parent cell that will be dividing, both daughter cells need to get one copy of each of the chromosomes. And because of the internal membranes, the only way for this process to work is to break down the internal membranes into really small pieces right before cell division happens, and then to reconstitute the membranes in the two daughter cells once the process has completed. So the membranes will be broken down and then afterwards reformed. Mitosis is going to be a process we talk a lot about, and this is the process of organizing chromosomes, then attaching them to opposite sides of a cell, and finally allowing them to divide among the daughter cells. And so this accomplishes the complex task of making sure that every daughter cell gets one copy of each chromosome and avoiding the situation where one daughter cell gets two copies of one while the other daughter cell does not get any copy. We'll come back and we'll talk much more about mitosis in a bit. So in cell division, our goal is going to be every daughter cell getting identical copies of the DNA. There's no change here if everything goes correctly in the information stored in the DNA of the daughter cells compared to the parent cell. At least in theory, you could substitute the DNA from one for the other, and it would make no difference. Because there's no difference in the DNA, it means that if the DNA gets replicated once during cell division, we will also have exactly one cell division following that to result in two daughter cells. So you can go through the math here. The math is basically as simple as we have one cell or one copy of the DNA. We replicate it, and so that means we now have a cell that has two copies of the DNA. But that's not a good situation. The daughter cells want to each have the same as the parent cell, and so we have to take those two copies, divide them into two daughter cells, and we are back to a situation where each daughter cell has one copy. Of course, what we got out of this process is that we now have two daughter cells. The organism has grown. Because the DNA will be identical in daughter cells compared to the parent cell, if the process that is occurring is mitosis, and if that process is occurring in a diploid cell, remember diploid cells have two copies of each chromosome, so we refer to that as 2n. A diploid cell then that is 2n, if it goes through mitosis, is going to make two cells that are 2n. And a haploid cell that goes through mitosis. So a haploid cell, remember, has one copy of each chromosome, or it's 1n. If this cell goes through mitosis, then it is again will make two cells, but now they are 1n. Nothing has changed. Now, in animal life cycles, haploid cells never go through mitosis. Or at least in mammal life cycles, they never do. This is not true in plants and in fungi. In both plants and fungi, there are parts of the life cycle where the entire organism is haploid, and its haploid cells are going through mitosis, making more haploid cells. In plants, there are also diploid cells that, like animals, go through mitosis and make more diploid cells. So both can happen. If we focus only on animals, then this will happen in mitosis. But in animals that we'll talk about, haploid cells will never go through mitosis. Let's consider the case of jackjumper ants, simply because the numbers are simple. This is an unusual eukaryote in that it has only one chromosome or one pair of chromosomes. Females in jackjumper ants are diploid, and that means that the females, since they're 2n and there's only one chromosome or one pair of chromosomes, then they must have two chromosomes. Males are haploid. Haploid, the root of this word is half. Diploid, the root of this word, or the prefix rather, di, means two. And so that's how you can remember that diploid is two copies, haploid is half, or one copy. So for a species that has only one chromosome or pair of chromosomes, the haploid number will be one chromosome. Let's now think a little bit more about the structure of eukaryotic chromosomes. First, let's emphasize that there's a lot of DNA in our cells. So every single cell in your body, or at least all of your somatic cells, have about 2 meters of DNA. That's long. If you imagine a meter is longer than I can fit on the screen here, double that and we're up to 2 meters. But remember also that your body has trillions of cells. And so if each cell has 2 meters of DNA, and you have somewhere around 2 trillion cells in your body, that means you have about 4 trillion meters of DNA. To give you a sense of how gargantuan that is, we're talking about something like the distance from somewhere around the sun out to maybe Uranus. So that is a lot of DNA to organize. Obviously, most of that organization is getting it divided into 2 trillion different cells. But then even within your cell that is only micrometers across, with a nucleus that's even smaller, still has to organize this 2 meters of DNA. How does it do this? First, remember that the nucleotides are not randomly strewn about. Instead, they are in a sort of linear pattern, but that linear pattern twists into a helix. And the helix is double-stranded because, as we've talked about, there is complementarity between different nucleotides. For example, C and G or A and T attached to one another. So we have a double-stranded DNA that winds itself into a helical form. Already that's going to help reduce the length. If you're not convinced by that, just take a string, hold it between your fingers and twist it, and you'll see that as you twist it, it will get a little bit shorter. The next thing that will happen is the DNA, which is represented by this blue tube, will wrap around a histone. A histone is a set of several positively charged proteins that are attached to one another. And DNA has a slight negative charge, and so it's attracted to the histones, and it wraps around in this 2 loops around each histone. And so you can see we've reduced the length of the DNA. This is much like if you wanted to organize a hose, you could coil the hose maybe around the center part of a hose reel. So that's our second step in organization. Now we have, we should say, this set of proteins is called a histone. The set of DNA around the set of proteins is called a nucleosome. So here's the word nucleosome. It's not synonymous with histones. Histones are the proteins only. Nucleosomes are the proteins with the wrapping DNA. Okay, so those are nucleosomes. The nucleosomes aren't just stretched out in a long chain. Instead, they form a coil themselves, and you can see that pictured here. This is sort of like if you had a phone cord and you continued twisting the phone cord, eventually it would form this higher level of coiling. And we will call these higher levels of coils, or the structure that's formed as a result, we'll call this the solenoid. Now the solenoid itself would be these long strands here, but they don't just form linear strands. Instead, they themselves form loops inside the cell. And we're not exactly sure what the structure of these are, but this is a good model to think about it. They can form loops that come back in to a central point, and this would form a structure called rosettes. And it's called rosettes simply because they look like flowers. This is not a separate chromosome. This strand is attached to the next strand back, and it's just that they're showing it separate to make it easier to see what the rosettes look like. And so this connected to this and then connected to a whole lot more of these would still just be one strand of DNA. So we have rosettes here. The rosettes can get even further compacted. And this happens in most cells only when the cell is about to go through some form of cell division. During cell division, there's a lot of potential for tangling because the DNA is moving around. So it's really important to get it as compact as possible. And so we are going to call this mitotic chromosomes. Mitotic. My spelling is terrible today. Or we can call it fully compacted chromosomes. And this will happen only during cell division. Let's just put this back the way it was. So this will happen during mitosis. We'll see that it will also happen during meiosis when we get to that chapter. So that is a little bit about DNA organization. Now, if we actually look at the nucleus of a cell, let's say by using scanning electron microscopy, as this image was taken, then what we can see is, so we see first the two membranes surrounding the nucleus. Remember, it's got a double membrane. And we can see a nuclear pore. This is an opening with proteins surrounding it that will allow proteins and RNA to move back and forth. More importantly here, we can see some areas that are really dark. This is areas where the DNA is really, really dense. And the term for those areas is heterochromatin. So this is the most compacted DNA. The DNA in the heterochromatin is not expressed. In other words, the information in it is not frequently used to make RNA with the RNA sent out to be the code to make proteins. Instead, so you can think of the heterochromatin sort of like when you really shove some clothes you don't want to wear into the back of a drawer. It's compacted, it's out of the way, but it's also not going to be used. In contrast to that is the euchromatin. And the euchromatin is more spread out, so that's why it looks less dark here. These white areas aren't really empty, it's just more diffuse DNA. Because it's more spread out, it is easier for the machinery that's necessary to attach to it to make RNA. And so we can get RNA that gets sent out into the cytoplasm where ribosomes will use the information as code to know what kind of proteins to make. So euchromatin has its genes expressed. Expressed means made into proteins. Heterochromatin is not expressed. Now, remember that our cells don't just have one chromosome. So we've talked about the different levels of organization in an individual chromosome like this. But human cells, for example, have 23 pairs of chromosomes, and almost all eukaryotes have multiple pairs, ranging anywhere from 1, as we saw for the ant example, the jackjumper ant, and then some have a lot of chromosomes. For example, this fairly simple, fairly ancient fern has 1,262 chromosomes. I'm a little bit dubious that anyone has counted them successfully, but that's a good estimate of its number. So the number of chromosomes does not tell you how complex the organism is, but either way, there are usually multiple of them. Here we are, humans, we have 23 pairs, which is the same thing as 46 chromosomes. So here's a karyotype. This is simply a visual representation of chromosomes, and this is the karyotype for humans. We can see these are next to each other because they have related information. One of these would have come from the mother, one would have come from the father, but these are not actually connected to each other. You can see these are each separate structures. And so if you count them, again, you can see the 22 normal pairs, and then our 1 pair of sex chromosomes, so that's 23. And again, because each one is a pair, that's 46 in total. 23 pairs. Now, in typical humans, we have either an X chromosome and a Y chromosome for males, or 2 X chromosomes for females. And we will talk later in the semester about the fact that this sometimes varies. We can have aneuploidy, especially for sex chromosomes, and it is survivable. So sometimes we could have an XYY, for example. But let's focus on the typical case. Most males are XY, and most females are XX. Now, if you think about what the karyotype of human eggs or human sperm would look like, they would not have all of these chromosomes. We will talk much more about meiosis in a subsequent chapter, but for eggs and sperm, then both eggs and sperm have only one copy of our DNA instead of two copies. So instead of having 23 pairs of chromosomes, egg or sperm would have they would have 23 chromosomes in total, not 46. So in this diagram, we are going to start thinking about what happens when chromosomes replicate. And what we have here on the left is the typical condition. This is when DNA replication has not happened yet. So we have chromosomes from mom, in this case in blue, and chromosomes from dad, in this case in red. These two chromosomes have very similar sequences of DNA in a similar order, and they have the same sets of genes on them. For example, there might be genes for anything from digestive proteins to pigments for hair color or skin color to pigments that make a neurotransmitter, or genes that make a neurotransmitter. And if there's a gene for a neurotransmitter right here on mom's chromosome, there's also a gene for a neurotransmitter right here on dad's chromosome. And if there's a gene for eye color right here on mom's, then there's also a gene for eye color right here on dad's. These chromosomes, in that sense, are the same. Now, there might be variation in exactly the copy of the gene each one of them has, but at least it's the same set of genes. Maybe mom has a gene for blue eyes and dad has a gene for brown eyes, but at least they're both eye color genes. So the prefix homo means same, and so homologous chromosomes they are the paired chromosomes that each have the same sets of genes on them. This is what the chromosomes look like before they divide. Just one copy of the DNA on mom's chromosome and one copy of the DNA on dad's. After DNA replication, we'll see that we end up with two copies of the information on both of the chromosomes. So mom's chromosome has replicated, and now there are two literally identical copies of DNA on each of the structures here. So again, if there's a gene for eye color here, there's also a gene for eye color here. But while mom and dad might have had genes for different eye colors, this one will be exactly identical to this one, all the way down to the exact sequence of A, C, G, and T on both copies. In other words, these are identical to one another. They are also attached to each other. So we need a name to talk about them. We will call each of the identical structures sister chromatids. Now, somewhat confusingly, we are still going to call this structure as a whole a chromosome. So this over here was a chromosome. Now it's replicated, but we still call it a chromosome. We don't start calling it two chromosomes until this divides and they are two non-attached structures. Now this region here that attaches the two chromosomes, let me re-say that more carefully. This region of DNA that is near the point of attachment is called the centromere. We'll see that the actual proteins, the green things here are proteins, not DNA. The protein that is in between the two is called cohesin. But the region of DNA in the area where cohesin forms is called the centromere. Now, this is one chromosome. This over here is still also one chromosome. It also is composed of two sister chromatids. And we still will refer to this chromosome and this chromosome as being homologous to each other. So our language about homologous chromosomes doesn't change just because they've both copied themselves.